So I just want to tell you what we're going to try to cover today and what we hope that you'll be able to walk out of this with. We hope that by the end of this workshop, you'll be able to clone, create new workspaces. You'll be able to upload data using Google Console. You'll be able to upload metadata to the data model. We'll talk about the data model later. You'll be able to amend and re-upload metadata. So say you want to reanalyze your data on a subset, you'll learn how to do that. We'll learn how to import a method from the method repository, how to launch an analysis, how to monitor all your analysis runs, do some really basic level troubleshooting, and then how to review results and reports. We also hope that you'll have a basic understanding of sort of conceptual parts of FireCloud. So we'll start with just a tour of preloaded workspaces and methods that are currently available. We need to talk some about administration of FireCloud because all this goes through Google Cloud. So there'll be billing pieces, accounts and buckets. We'll talk about the data model, help you understand that. We'll talk about method configuration basics, tasks, workflows, and WDL, which is the workflow description language that we use in FireCloud. We'll talk about TCGA controlled access data and dbGaP authorization, because as you know, all of TCGA data is loaded here. So it's available for use with proper authorization. And lastly, we'll talk about proper handling of TCGA controlled access data. So hopefully we'll cover all of this. All right, so what exactly will we be doing? We're going to try something new here. You're sort of guinea pigs. I hope you don't mind. And what we're going to do is every time we present a piece, we're actually going to have you do that hands-on. And gradually, we're going to build through the whole exercise. Basically, you guys will execute an analysis run by the end of this. And exactly what analysis will you run? This is our affectionately known as the GDAC hairball. These are all the steps of the GDAC pipeline. And those in purple are the ones we'll actually execute today. So that'll consist of a clustering algorithm that will run on two different data types, mRNA-seq and RPPA. Then we'll aggregate those clusters. And lastly, we'll run a program that correlates clinical versus the molecular subtypes. And hopefully, along the way, you'll learn all the FireCloud basics and be able to do this yourselves later. So that's sort of our goal for what we want to cover today. Okay.
Okay, before we start, or we are starting, so we are kind of going back and forth between describing sort of concepts and then hands-on exercises. Please feel free to interrupt us at any time if you have any questions about some of the concepts that we are presenting. Please raise your hand and we'll go ahead and try to clarify that for you and then clarify it for the entire group. Okay.
Okay. So FireCloud is a cancer genome analysis platform that runs on the cloud. As probably most of you know, it was modeled after Firehose. Firehose sort of hit the maximum of its expandable life in the form it's currently in. So FireCloud is next generation. Currently, FireCloud runs on the Google Cloud platform. FireCloud is designed for three main user groups: analysts, production managers, and tool developers. I just want to be really clear. What we're giving you today is really the analysts' workshop. We will also do production manager and tool developer workshops. Those will be later, so keep an eye out for those. But this one really is specifically for analysts. And before I go any farther, I meant to do this first. I want to introduce our team. I'm Megan Hanna. This is the Cloud Pilot team over here. This is Chet Berger, Principal Architect. Eddie Salinas, Software Engineer. Jason Neff, our Technical Writer. Alex Bauman, our Engineer. He's over there. So any questions, just pester us, okay? We're also going to tag team it and trade on and off, so hopefully this will work well. Okay, so pre-populated workspaces. What's in there now? Okay, so this is sort of our starting. This is where we're starting with FireCloud. We have three basic types of workspaces. We have our tutorial workspaces. Tutorials contain data and methods, right? So you can take a tutorial and actually, execute the run. We have a lot of data-only workspaces that contain TCGA data. TCGA data has two flavors: controlled access, which contains things like snip arrays, anything that can be identifiable back to the participant. That will contain BAM, snip array files, birdseed calls, a host of data types. And then we have open access TCGA data. All of this is publicly available. You'll see expression, copy number, clinical data, a whole lot of varieties of data there too. All of these are organized in the workspaces by tumor subtype, okay? We also, the last set of workspaces we have that are preloaded are best practice workspaces. These contain our sort of best practice pipelines and workflows. The way we're trying to create them is so they too are just ready to run. All you need to do is load data. Okay, so that's what we have now. Some of these are controlled access only, and you're going to have to do dbGaP authorization, and we'll talk to you about that only later. But right now, you should be able to see all of these in your workspaces now. Okay, so let me just show you what that looks like. All right, so I'm going to log on to FireCloud. Oh, and one other thing. Right here, this page right here is our landing page. Here, we have our forum. Please bring all your questions to the forum. Okay, we monitor the forum every day, and the team here will help answer anything. If something is technical or engineering, we'll triage it and it will move up to the engineers. Also, any comments, any requests, please funnel everything through there. In addition, we have a user guide that's pretty extensive, so feel free to look around, see what you need. Again, any kind of comments, let us know, post it in the forum. We need to learn from you. Okay, so I'm going to enter FireCloud here. I'm already signed into my Google account. So here we are. And I'm just going to filter here on open. And here we can see the open access workspaces. You can see that they're arranged by tumor subtype. So we've got POD, DLBCL, COAD, ACC. These are all the TCGA subtypes. I am authorized for dbGaP. So I'm going to show you controlled workspaces. There's the controlled workspaces. Note they have a restricted tab right there to let you know that you're actually entering controlled access workspace. Data that's derived from controlled access workspaces may also be controlled access data. So we also have ways to protect those workspaces that will actually inherit the protection. And you'll learn about that later too.
Megan?
Yes?
Is controlled access and dbGaP, are those two things tied together, or could my users have controlled access that's unrelated to?
In this case, we're talking specifically about TCGA. Eventually, FireCloud probably will host other data types that may also have controlled access data. But for now, we're only loaded with TCGA data. But yeah, eventually we'll hold others. Okay, and last thing, I'll show you best practice. Let's see, here are some of our best practice. Here's some of our best practice pipelines. There where you see mutation calling pipeline, and we'll talk about what's in those later also. And then we also, lastly, we have tutorial workspaces. Okay, so what's available today? I know this is the first question everyone's going to ask, so I figured I'd just get it out of the way now. This is what we've got now. Loaded, you could access all of these tools right now. We have our mini mutation calling tutorial, which includes it runs on mini BAMs, and it's also a little truncated mutation calling that just has contest, mutect1, oncutator. We have a handful of GDAC analysis tools. That's the cluster analysis that we're going to run today, and a couple of other ones. Our best practice mutation calling workflow, this is actually one of 4 parts. We have 2 of them available now. One is the calling module, which has QC copy number, cross-check fingerprints, contest, and Picard metrics. The second one is the actual calling workflow, mutex 1, mutex 2, encodetator, and variant effect predictor. So that's what's available now. Under construction are the following, the 2 remaining best practice mutation calling modules, the copy number one, which has absolute deranger, RE capseg, and allelic capseg, and then the filtering module, which will have orientation bias filter, which probably most of you know as OXO-G or OXO-N. There's a couple of OXOs for artifact detection. D10, a panel of normal filter and a realignment filter. Also, we've got more GDAC methods coming in. We also have the GTEX pipeline. That's almost ready to roll out. So hopefully in a few weeks we'll have that ready to go. We also have a bring your own panel of normals. And panel of normals we can discuss elsewhere, but that's something that will be very useful for a lot of things. And coming, we haven't quite begun, are things like MuteSig and PhiLogic. Anything you need to do your work, any method or program, let us know. And if it's something that it's within our ability to get into FireCloud, we will. And based on how many people request it, we'll prioritize it higher. Any new tools, we can help you get into FireCloud. So let us know, because sort of this is the important part here. And this is sort of a #1 priority for us now, getting tools, methods, and workflows up into FireCloud. Okay. I'm going to pass this over to Jason, who's going to do he's going to walk you through the mini mutation calling tutorial. So you'll see all the basics, and then we'll actually begin to do some of those.
Can everyone hear me? Okay, great. Also, just wanted to give you a quick update on the account activation. I think we're still working on that right now. So if you happen to arrive to the workshop just a little bit late, we wanted you to log in to firecloud.org to see if you are activated. If you are activated, you'll be able to see workspaces in FireCloud. And if not, you can just let one of us know, and we'll help you get your account activated for the workshop. So now what we're going to do is we're going to demonstrate how to launch an analysis using the BroadE mini mutation calling workflow. This will give you a basic overview of some of the concepts that we're going to cover in this workshop, and it will also show you how to actually go through one of our tutorial workspaces that we have available in FireCloud. So when you first log into FireCloud, you'll see the workspace list. From this page, you can view and search for any workspaces that you have access to. Once you have an active account, you'll be able to see the tutorial and best practice workspaces, as Megan mentioned. So what I'm going to do to try to find the mini mutation calling workflow, I'm going to use this search function in FireCloud, and I'm going to type in mini. And if you click on that workspace, it'll take you to the Summary tab. Here it has a brief description of the workspace, and you'll see there's also a link to a tutorial that we have in the FireCloud forum. And what I'm going to do before I launch this analysis, I'm going to clone the workspace. This is a concept that we'll cover later in the workshop. Okay, so if you navigate to the Data tab, here you can view the workspace data model, which includes entities and attributes. For example, you'll notice that there are pairs, pair set, participant, and sample available in this workspace. If we click on the Method Configurations tab, here we'll see that this workspace is pre-populated with a method called mini mutation calling. if we click on this method, we'll be taken to the method configuration. It's another concept that we'll describe in this workshop. But most importantly, at this point, I want to show you how to launch an analysis. So by default, this method is run on pairs. You can also run this on a pair set, which would run it on both of the pairs in this workspace. To do so, you would just select the pair set, and you'd want to type in this dot pairs. And then you would click launch, and the analysis would be run on both pairs in this workspace. For the sake of time, what I'm going to do is just run on a single pair so that we can actually have results to show you later in this workshop. Okay, so later on in this workshop, I'm going to show you the results of this analysis. Another concept, I just wanted to briefly preview the method repository. In the method repository, you'll notice that there are also preloaded methods that contain the tools in FireCloud. You use these methods to run an analysis in FireCloud.
Can you explain a little difference between methods and method configs?
Sure. So also loaded in the method repository, we have method configs. These contain inputs and outputs that already have all of the references that you need to launch your analysis. Chet, is there anything else that you wanted to add about method configurations?
Yeah, okay, so when we define tools, what we do is, there are workflows that are defined in this workflow description language called WDL, and we explicitly declare what the inputs and outputs are. And the inputs and outputs might be files, and the inputs can also be literal values and integers. Rather than explicitly declare when we run a tool what those inputs and outputs are, we provide the option of binding those to attributes that are in our data model. So for example, if you have a sample entity and there is and say that sample is a primary tumor and there's a band associated with it, there'd be an attribute associated with that sample, a band. And what you would do is in the method configuration is what will go ahead and link a workflow input to a value that's defined in the data model itself. And the advantage of doing this is that you can potentially go ahead and suppose you have 1,000 samples and you want to go ahead and run an analysis on each of the bands for all those samples. What you would do is you would go ahead and write a method config that was targeted at samples, and you go ahead and run that against a whole collection of samples. And when you want to specify what the input parameter for the BAM file is, all that you would need to do is say, for any of the samples that we're analyzing, you go ahead and get the path or whatever to the file that's associated with that sample. It's one of the annotations off of that sample. It'll become much clearer when we go ahead and actually walk through and we'll take a look at some of these method configs. How many people here are familiar with Firehose and Firehose users? Okay, so many of you who are about 20% of you are familiar with Firehose. These method configs are analogous to the task configs in Firehose. Yes?
Before we proceed, I think many, if not most folks in the room received an error when attempting to access the method configuration in the mini patient calling workflow. Okay. I believe you said that we should be going along, but I'm not sure if that's going to be a problem later on. Or
Yes, for this purpose of the workshop, we just wanted to demo how to launch an analysis. So it's possible that you just do not have access to that method, but there will be other methods in this workshop in which you will have access to, and we'll actually have you
You all
run along.
should have access to the tutorial. Did that get published? The mini retracement column?
I believe
Method we don't have access to.
Okay.
Yeah, so maybe there is an access control
Okay.
issue with that.
Yeah, we'll
Yep.
check on that.
But that was primarily just to give you a quick look at this. Now we're going through the GDAC pipeline, or a portion of the GDAC pipeline. And we know that you have access to all of those methods and all of those situations.
Perfect.
Okay. All right. Before we, are we done with this portion of it?
Yep.
Okay. So what's next here? Oh, logging in.
Logging in. Oh, yeah, it is. Sorry. So you all know you need a Google app, a Gmail, but obviously you're all in, so I think we're good there. And everybody, is there anyone left who can't get in? No? Good. Oh, one person, two people. Eddie or Jason, you want to go see who those people are? Let's get you guys in. Okay. One other thing I just wanted to say, at the bottom, there's a link at the bottom of the sign-in page. That link takes you to the data use agreement for TCGA data. So if any of you are using TCGA data, please read this. This is what it takes you to. It talks about, basically the data use agreement is based on the patient consent, so it's important to be familiar with it. Okay. We're going to talk about Google Billing and Google Projects.
Okay, great. Am I on? All right. Okay, as we said, FireCloud runs on the Google Cloud platform. It's a fabulous environment for running genomic analysis on the cloud. There's one problem with it, and that's that you still have to pay. And now what we're going to do is talk about how you actually go ahead and handle the chargings for it. The way that the Google Cloud environment operates, all of the charges, both storage costs and compute costs are charged to a Google project. And when you create a workspace in FireCloud, either through cloning an existing workspace or creating a new workspace de novo, you have to specify what the Google project is. And that's the only time you can go ahead and specify what the Google project is. Once it's set in stone and you can't change it. And what that means is that any work that's done in that workspace, that any compute costs incurred in that workspace are going to be charged to that Google project, and any data that is uploaded to it or generated and has to be stored in the cloud, the storage costs are also going to be charged to that Google project. Okay. Google project is linked to a Google billing account. And for users outside of the BroadE, that Google billing account, there may be a credit card behind it. For those of us in the BroadE community, what we do is we're going to associate, there's actually just going to be a single Google billing account that the BroadE uses, but we go ahead and associate each project with a cost object. So when you want to start using FireCloud and you want to charge your work to a particular cost object associated with the grant, what you need to do is to go ahead and contact us, send us an email, and make a request to go ahead and create a Google billing project, a Google project, and provide us with the cost object for that project. And we will go ahead and set that up for you. Now, you can have there isn't a one-to-one relationship between Google projects and workspaces. For example, you might have a PI, and he goes ahead or she goes ahead and sets up a Google project, and that Google project is associated with the cost object, and it's intended to be used by all of the researchers in his or her lab. And the PI will go ahead and send a request to help@firecloud.org, and we will go ahead and make sure, and that request will be to make that Google project available to all of the users that he or she specifies in the lab. Okay. For this workshop, we went ahead and created a Google project called BroadE-FireCloud-BroadE. You should all have access to that. And we'll be able to verify that you do have access to it in a moment. All right, next mobile projects. All right, next, okay. So as I said, when somebody creates a workspace, that workspace is going to be associated with the Google project. And also, when you go ahead and create a workspace, a dedicated bucket is assigned to that workspace. And a bucket, you can view it as a file system, or it's a place where you can store data objects. And as we said, the Google project is going to be linked to that bucket. So any storage costs that are associated with that Google project are going to be charged, are associated with that bucket, are going to be charged to that project. And the project is then linked to a Google billing account. We had talked about, we touched briefly on workspaces and a data model that has entities like pairs and participants and samples and attributes associated with those. For example, a BAM file or maybe a clinical file. Those files are within the workspace and in your data model. If you go ahead and take a look at those entities, and we will be doing that, you'll be able to see that there are pointers to files in buckets within cloud storage. Those files may point to the bucket that is directly linked to the workspace, or they could be pointed to some external bucket. So for example, if all of the TCGA data is stored in, we have two buckets. We have an open access bucket and a closed access bucket. And if you were to go to any of the TCGA workspaces, open or closed, and take a look at the attributes and see that they point to files, you'll see that there are references to one of those two buckets. Each of those buckets, each of those workspaces, TCGA workspaces, also has a dedicated bucket, but there's no data in it. Okay. Any questions at this point?
So important to know that you won't be paying for all of the TCGA data
Yes,
every
right.
time you use the system.
Right. Okay. Yes.
Do you know anything about external users billing?
Yeah. So what we're going to, for external users, if they want to provide a credit card, then they would go ahead and use the Google the Google console to go ahead and create a billing account. And then they would go ahead and in their request to set up a project, they would provide us with that Google billing account ID. But we know that researchers in a lot of institutions don't have credit cards. And so right now what we're doing is working on we're working with a Google reseller who's going to try to make that easy for us. And so that means that whether you're at MGH or any university, you go ahead and contact them, and they would go ahead and set up the Google billing account for you with whatever sort of payment mechanism that institution wants to use. And you would then go ahead and provide us with that Google billing account ID.
That Google reseller is part of Google?
No, it's a separate company whose job is to try to make Google's billing infrastructure a little bit easier to use for people. Okay, but that isn't in place right now. It's being worked on. But for all of you who are in the BroadE community, it's pretty simple. All that you do is go ahead and provide us with the cost object. Okay. Where are we now? Okay.
You want to talk about charges for cloned workspaces
Yes.
and
Okay.
shallow
So quite often the way people are going to operate is, suppose I want to do work with a particular cohort from the TCGA data set. We have those workspaces available. They're all locked. You aren't actually going to be able to do work in them. You only have read access to them. But you can go ahead and clone those. And when you go ahead and clone that workspace, you're going to make a copy of that workspace. You're not going to copy all the data. You're going to copy the data model that has a list of all the entities. And as we said before, the attributes associated with those entities can contain references to files. Those files are going to go ahead and point to the TCGA bucket. So when you clone a workspace, you aren't going to have to pay for that data. We're doing something called the shallow copy. But when you clone a workspace, you're creating a bucket associated with it. And any analysis that you run within that cloned workspace, the output files are going to get written back to this bucket. And that's what you're going to be paying for. Okay? And you'll also pay for any of the computational costs for running tools within that workspace. And that will be charged to the project. Yes?
How much are these costs now?
I don't know.
I think storage is like $26 per terabyte a month. So storage is pretty cheap. It's actually considerably less than it is here at the BroadE right now. The compute, there's a whole range that you can look at their price list. That'll probably help. Eventually, we'll have little widgets that will actually tell you what a workflow will cost to run based on estimates, but we're not there yet.
Yeah. But if you're doing work with relatively small cohorts, a couple of hundred, and you run a workflow that maybe could even potentially take hours or days to run, you're still not going to rack up a very expensive bill.
Yeah, we've run a couple of pan-cancer tests, just running a lot of data, and it didn't cost us more than $300 or $400 for the month. So, yeah.
Has there been any work on using or adding kind of workflows associated with VCF and with VCF manipulation and analysis?
Not yet, but let us know what the tools that you need are, and we're certainly open. Either we can help you get set up to put them in yourself, or if there's enough interest, we'll work with you.
Okay, all right. Next slide. Okay, so this, we went over this already.
Okay.
Okay, so now we're going to go ahead and clone our first workspace. Okay, so what we're going to do is let's go take a look at the workspaces. And we're interested in the workspaces associated with this cohort, adrenocortical carcinoma.
Has everybody seen?
Everybody should follow along here.
Yeah.
And and you can see that there are two data workspaces associated with this. One is open access and one is controlled access. And notice that the controlled access has this little visual cue here to indicate that it's restricted.
How can we make the font a little bit?
You can't see it, okay. It's good for me too, because my eyes aren't very good. So.
How's that? Is that better?
Is that better? Okay, great. All right, so let's quickly jump into the open access workspace. And let's just take a quick...
Pick ACC.
Yeah,
because
ACC,
it's the
open
smallest
access.
of the TCGA data sets.
Okay. And we're going to go ahead and take a look. I just want to sort of talk a little bit about another quick view of the data entities. There are participants, samples, and sample sets. If we go ahead and take a look at the samples, each column here represents an attribute. And if we were to go ahead and, And if we were going to go ahead and take a look at this, we'll see that this is I was referring to there being an open access bucket. This file right here resides on this bucket, and this is the identifier of that file on that open access bucket. And if you want, you can go ahead and download it by clicking open. But in fact, there's no reason to go ahead and download these files because you're going to be doing all of the analysis in the cloud itself. All right. And the tools that we're going to be running are going to be, for the purposes of time, we had talked about this being a system that is highly scalable, where you can go ahead and have 184 or 1,000 samples and run one analysis across that entire collection of samples. And you'll go ahead and it'll be run, or in parallel, it'll go ahead and run that analysis on all thousands of samples at once. And since we're running on the cloud compute infrastructure, it's highly elastic, so you'll be able to get all the virtual machines that you need in order to run that analysis. But for the purposes of this workshop, we're going to focus on analyses that run on a single entity, and that entity is going to be a sample set. And we're going to be, right now, the sample set entity, there's a single entity, it's called ACC, and its only attribute is a list of all the samples that are associated with it. We're going to be adding additional attributes, additional files to this system, and then we're going to be running the small portion of the GDAC pipeline on this. Right now, there are no method configurations, and there's nothing to monitor. over the course of this workshop, we're going to be sort of filling these things in, and you'll actually get some live data that you'll be able to work with. But the starting point is going ahead and cloning this. Now, don't clone it yet, because I want you to. So what we're going to be doing is all of you, if you click on clone, you should be able to see that you have access to this Google project, BroadE FireCloud-BroadE. So you're all going to go ahead and clone this workspace under that Google project. Okay. Now, what uniquely identifies a workspace is the combination of the project name and the name of the workspace. So if all of you were to go ahead and clone this workspace using the existing name, there'd be all sorts of collisions and we'd start having problems. So it's important that each of you go ahead and clone this workspace, giving it a different name. And what we'd like you to do is go ahead and clone this workspace, appending to the end of the name your user ID. So for example, we're working under Megan's user ID, and I'm just going to add M. Hannah to this. Okay, so you should all do that. And then you click OK. And we have all 50 of us trying to clone a workspace at once, and there may be some delays associated with this.
Sorry.
Yes?
So the fact that there are these collisions, if we all use the same name, means that when we clone a workspace, it's available for everyone?
No, I think what will happen is if there's a name collision, if it attempts to go ahead and clone a workspace, and that workspace name already exists, you'll get an error message.
Okay.
So the first person who got the name and cloned the workspace will be the one who owns that. Here we go. So here we have, this is the new workspace name, and you can see that the data is identical. Okay. How many of you have been able to get to this spot?
Good.
Great. Okay. All right. Now we're going to talk about workspace access controls. Associated with each workspace is an access control list, because this platform is intended to go ahead and promote collaboration. And there are lots of different ways of collaborating. One way to collaborate is to go ahead and create a workspace and then provide your collaborators access to that workspace. And you can provide different levels of access to a workspace. The kind of the lowest level of privileges you can grant somebody is reader level privileges. And so if I create a workspace and I grant a collaborator reader level privileges, that means they'll be able to see that workspace in the listing of workspaces that are accessible to them. They'll be able to enter it. They'll be able to view the contents of it. They'll be able to download the data from it if they want. And they'll also be able to clone it. But they won't be able to make any changes to it. They won't be able to upload data. They won't be able to create things called method configurations in that workspace. And also, most importantly, they won't be able to go ahead and run any analysis, which would then potentially change the data in that workspace.
Chip?
Yes?
So readers who can download files, that means they can cost you money.
Good, yes.
That's good.
Yeah. And in fact, so when you should choose your collaborators carefully.
Wisely.
Okay. Right? Yeah, and that's actually an issue also for these TCGA open access workspaces, and the closed access workspaces that have these enormous spam files. Those TCGA workspaces, we pay for them. We pay for the storage costs for them. Data egress charges can be quite expensive. And unfortunately, Google right now doesn't provide us with a mechanism for blocking downloading of data.
Great. And another thing that's really important to note is Google is a business. So the way it works is different from the way we're used to working. Upload is free. Storage is cheap. Download really costs you. So they want you to put the data up there and keep it there. So just be mindful of that, like downloading a BAM or a set of BAMs will cost you a lot.
Yeah, and we are going to be monitoring this. And so if we find FireCloud users downloading and incurring very large data egress costs, we'll be contacting you.
Yeah, and the thing is, there's actually no need to download TCGA BAMs or any TCGA data, because it's all there for you. So,
Yeah.
you know, I mean, there's a few reasons if people want to do realignments and they're creating new BAMs and then you want to download those. But those are sort of edge cases.
Yeah, you had a question.
Yeah, just a quick question about the Google Cloud. My understanding is that transferring data internally within Google Cloud is free.
Yes.
So I know that at the Broad, everyone's used to like working in like a traditional Linux environment where all of our files are on the the Unix file path.
Right.
So I'm curious for like end users who are used to accessing things that way. Is there a way to run your stuff on the Google Cloud, but then log into, say, spin up a node on their cloud and view the data there instead of downloading everything?
I don't, there may be. I mean, but one of the kind of models that people are using is a group may go ahead and create their own dedicated bucket. And they'll put all their files in that bucket. And then they would go ahead and create workspaces that would reference files in that bucket. And so they would be available to everyone within that group. But in terms of sort of launching a VM and then sort of the way that that this platform operates is when you actually run tools and you want to run the tool on the file, the Google system does something called data localization. It'll go ahead and copy those files from the bucket onto the virtual machine that the tool is running on. And then when the results that are on that virtual machine are completed, it'll go ahead and copy those new generated files back up to the bucket. But I don't think that there's any way to sort of establish a virtual file system that immediately points to files in the buckets themselves.
If it's just data, like looking at what's there and sort of doing your kind of LS and things, GSUtil, the command line tool works a lot like, a Unix prompt for doing that sort of stuff. It's when you want to open the file and look at its contents that that's
But if you use GSUtil locally, you will be pulling it down. If you want to use that in a VM in Google infrastructure, will charge you.
Yeah.
So just to LS.
LS probably not, but cat, yes.
Yeah, I'm saying as long as if you're just like saying, oh, what files do I have, and
Yes,
yeah, it presents sort of a pseudo file system.
You gotta be careful what you type.
Right.
Yeah.
Right. Okay, let's see. All right, so we're going to do, and also when you create we were talked about reader level access. Writer level access controls are all reader level access controls, plus giving somebody the ability to go ahead and write to that workspace. And that means giving them the ability to go ahead and upload data to it, upload data to the bucket itself, and also to go ahead and run analyses. And then there are owner level privileges, which are reader and writer level privileges, plus the ability to edit an ACL. Okay. Now what we wanted to do was an exercise, which is kind of like get to know your neighbor. So each of you have gone ahead and cloned the ACC workspace, and it's under your own name. Turn to your neighbor, introduce yourself, find out what their Google identity under which they're registered with FireCloud is, and provide them with read access to your workspace.
All right, was everybody able to share and also open their shared workspace?
Okay.
Yes?
All right, so this is an example of how we've designed FireCloud to make it very easy to collaborate with your users. But once again, you got to make another mode of collaboration is rather than say giving somebody, well, one mode is giving somebody read access, and then that allows them to go ahead and clone that workspace, take a snapshot of it, and then work within their own copy of your workspace. Just remember that the references for the cloned workspace will point back to the bucket that was originally associated with the parent. And that means that if there are any changes in the in the parents' files, that means the clone workspace will go ahead and be affected by those changes. What we do plan to do at a future date is, we had talked about cloning being a shallow copy. We also want to go ahead and implement a clone that involves a deep copy, where you actually copy all the files to the new workspaces bucket. And that way, you actually get a frozen snapshot of the workspace that you cloned, and then you can go ahead and do your work within it. Yes?
So if a workspace is cloned, can the owner still delete it?
Yes.
So what would happen to the
Well, what will happen to the clone is that means that since the data model is referencing files that may be on the bucket of the parent workspace, then those references will no longer be valid. Yes?
And with the shallow copy, the egress charge for pulling down a file that
it'll be charged to the parent because those files reside on the bucket of the parents. Okay. All right. Okay. So this is kind of one mode of collaboration. A closer mode of collaboration would be to go ahead and give somebody write access, and so both of you are working within the same workspace at the same time. Right. Okay.
Okay. All right, so now I want to talk about loading data and metadata. I'm just going to say this, and then I'll get to talking about the data model, but metadata populates the data model, and I'll just hold on that for a second. The way we load data, there are two ways. We can load data via Google Console, or we can load it through GSUtil that Brad and Alex were just referring to. GSUtil is a Google command line tool for loading data. Probably a lot of you will end up using it. We didn't walk through that today because everyone would have had to download it and get it up and running on their environments, and we didn't want to have to get through that today. So we're just going to do Google Console today. If you look at your summary page, you see a Google Bucket link. Let me go there. So here I'm in my cloned workspace. I'm going to click on my Google Bucket link, and it's going to take me directly to the Google Console. So here's my bucket. You can see there are no objects in my bucket. Sorry, I'll make this bigger. No objects yet. What we want to do is we want to import one of the files that was in your workshop materials into your Google Bucket. Okay? So what I'm going to do is I'm going to upload a file. So here's the file we want, the one with the really long name, ACC, protein expression, blah, 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 data.data.txt. Upload that to your Google Bucket. And there it is. You should see it in your bucket. Now, while you're there, so now let's go back to the workspace. Notice you don't see anything. You can look at sample set. Oh, I should tell you what this sample file is. This sample file is a sample set file. It's all the protein expression from the ACC cohort combined into one file. That's what this data set is, okay? It's from the open access bucket. So next what we need to do is we need to revise our metadata files and upload them so that in the workspace you have that connection between the file and the file location. So that's what we're going to do next. Let me just see where we are. Oh yes, okay, we did that. Okay. So now I'm going to take 1 second, I'm going to talk about the metadata files. So uploading the data's metadata to the workspace populates the data model. Metadata must be in a specific format, and that's in TSV files that have specific entity types. So there'll be participant, sample, pair, participant set, TSV files. Each of those will have this required first column header here, entity, colon, participant ID, et cetera. And these files must be uploaded in a specific order. That actually is important. So now let's just talk about what exactly is the data model. The data model is a framework that captures and formalizes entity relationships. The method configuration then binds the data model to the workflow. Each method configuration is targeted to a particular entity type, and we call this the root entity type. In this case, we're loading a sample set data file, so the root entity type will be sample set. Okay, so the data model starts with a patient, right? In this case, we call it a participant in our diagram there. Because they're patients, they have some sort of a sample, DNA comes, gets sequenced, whatever. In our case, working in cancer, it's a tumor. So that is a sample. See the sample right there? In cancer, we routinely work with two samples, tumor and normal. So these samples constitute a pair. Oops, sorry, we'll go back. Samples constitute a pair. Then, if we want to analyze an entire cohort, we can move into a pair set, a participant set, a participant set, or a sample set. So one thing that's really important here is that currently in FireCloud, what we're using is the TCGA data model, which is based on pairs to a sample. There's a lot of complexity in that, but that's fundamentally the data model we're using now. The way we built the system is it's extensible to other data models. So for example, working with trios, working with germline data, working with samples that have a time course. All of those data models will be extensible. And at some point, as soon as we're able to develop that piece of it, you'll actually be able to select your data model. For now, we're working with the TCGA data model, which is the one we have right up here. It's actually quite adaptable for most needs. If you need help adapting something you're trying to do analysis on, just come to us and we'll help you with that. So that's the basic data model. So what does this look like? Right here, we have an example. Here we have an example TSV file, just the first 3 lines. This is participants, so these are the participant IDs. Then here we have participant-specific information. In this case, tumor type, gender, and age. This next column gives the file name and, importantly, the Google bucket address. Okay, so this is sort of the piece that links together your data and the workspace. So if we actually go in and look at some of these, we're actually going to look at the ACC ones. This is what a participant TSV looks like. So there again, we have participant ID, and we have the participant-specific information, and our standard first column header right here. We can look at sample. And here you begin to see the linking. There's our sample ID. Next to it is the participant ID. Next is the sample type, in this case TP for tumor primary and NB for normal blood, a TCGA sample ID. And this last column here describes a clinical file. So we have the file name and we have the Google bucket address there. Note the critical first few things, GS : / and then the bucket name.
Is there any, I see.
Oh, sorry.
Is there, can the Google buckets have hierarchical directories and like that?
Google buckets are actually flat.
They are flat, but you can go ahead and use slashes to go
Right.
ahead and sort of make it appear as though it's hierarchical.
Yeah, when you go into the Google console, you see the directory, they show up as folder.
Yep. Okay, so we looked at sample, and again, here's pair. You'll see the same sort of organization, participant ID, and in this case, the case followed by TP, and the control followed by NB. Okay, so remember that TSVs have to be uploaded in a specific order, and now you can sort of see why you've got to upload participant first in order to hang any of the rest of those entities off of that. Okay, so the next what we're going to do is we're actually going to upload a TSV that holds 2 data types into your cloned workspace. Oops, sorry. All right, so I'm actually going to go into the workspace. So now let's go into your workspace. And you see participant, we just looked at the data, sample and sample set. Sample set is where we're going to upload this data set. So click on import data, import from file, choose file. And now what we're going to upload here is sample set underscore set underscore entity underscore ACC dash GE dash clin dot text, okay? Sorry, that should be bigger too, shouldn't it? Oh well, can't make that bigger. Okay, I'm going to open that. Here you get a nice preview. This is actually quite a convenient feature. I can preview my data. I see I have gene expression and clinical. These are the 2 data types I'm going to upload here.
One thing to note is, very quick,
Sorry.
we aren't creating a new entity. What we're doing is updating the existing entity. We're adding additional metadata, additional columns to that.
Okay, so now if we look at sample set, we see that we have 2 more data types here. Here we have the gene expression file and a clinical file. I can actually look at these and open them. And here we have the clinical data for the ACC cohort. Another way that actually is really convenient to look at information is you can click on this little, I don't know, widget there, and it's actually easy to filter. This is important when there are a lot more entities than there are here. But for now, we see that we have our ACC, our gene expression, and our clinical data loaded here. Okay. Okay. Next, we want to add the protein expression. So you're actually going to have to edit your TSV file. So if you look at your, and there's 2 ways you can do it. You can do it the lazy way, which is fine. I'm amenable to that. Or you can do it the hard way. What we need to do is we need to add the protein expression file and the Google address from your bucket, okay? Right now, the 2 files that I added, the gene expression and the clinical, are located in the open access bucket. But you guys uploaded the protein expression file in your own Google bucket, right? So what you need to do is you need to edit the TSV to have an additional column. The column name is protein expression, cap P, cap E, no space. And beneath that, you're going to have GS : / and then you need to copy your Google bucket address and then add the file name. Does that make sense? I'm going to do it right now. I like to do this in Excel. So I am going to open my file. Hold on, I need my file first. I'm going to open my sample set file. I'm going to copy it. I'm going to put it into Excel, sorry, and again, it's very small. Oopsie, sorry, that was. Did that make sense, what I said? Sorry, download. Sorry, we're going to do this one. Okay, so here we can see, that's probably too small for people, isn't it? How's that? Is that better? Okay, So here we can see our gene expression, our clinical data, and again, you can see that we have the Google bucket addresses. So now I'm going to add a third column. I'm going to call this protein expression. And following this, I'm going to do GS://. And now I'm going to go back to my workspace and I'm going to get my Google bucket address. So from your summary tab, you can copy your bucket address, add it in. Now we need / and your file name. I'm actually going to copy it here, but you can copy it where you want. Okay. Now I'm going to save this as a TSV. Tab delimited will do. Oh, I should give it a name. Let's see. Added PE. We'll just call it that. Okay.
Just cancel it.
Yeah, that's all right. It should be saved. Okay. Now I'm going to go back in. And go back into the data tab. I'm going to go to sample set. I'm going to go back to import data. Import from file. Now we'll see whether I saved it properly. I didn't. Sorry. Very good. Glad someone's catching my errors. Okay, what did I do? Try that again. Sorry. Let's see.
I think some of us had trouble accessing, or it looked like we didn't have access, but then I realized like if you're signed into multiple Google accounts,
Yes.
you can have issues, so it might be worth.
Right.
Yeah.
Yes,
If you're running into
that's correct.
you can log out of your other Google account, or what you can do is open up an incognito window and log into FireCloud in that incognito using the Google ID that you registered on the FireCloud, that you registered FireCloud on.
We should add that in future workshops.
Yes.
Okay, so I'm going to add my file, open it now. Again, I can preview it and I can see that I have gene expression, clinical, and protein expression now. And I'm going to upload, and this will overwrite the previous metadata. So now if we go into sample set, we can see that we have all three. Does everyone get there? Yes? No? Anyone need help? Anyone else?
Did you save from Excel as a TSV?
I'm sorry?
How did you save from Excel as a TSV?
I saved it as a text.
Oh, as a document.
Text is fine.
Okay.
Yeah. All right, guys, how'd everyone do? Did most people get through this? Yeah? Okay, good. Any questions? Just, I'm sorry? Okay, and I just wanted to say one other thing. Remember that we cloned this from a TCGA workspace, and if you remember, my sample list didn't have this many things on it, and that's because this associated on the sample tab are all the sample-related files that were in the TCGA open access bucket. And an easy way to review these is using the little widget on the side, and you can actually see all of the files easily and filter down to ones you want to look at. So I just thought I'd point that out because this is a good place for it.
Okay, the other thing we just want to mention is that FireCloud is a new system, and we will be making the user experience easier over time. But right now, we just want to get the kind of base system up and running, and so that we can go ahead and run these analyses at scale. We understand that this work around having to go ahead and update your TSV files in an Excel, while you as analysts are probably very familiar with doing this, we realize it's still kind of a pain, and we will be trying to make the system easier over time.
Okay. Let's see. Where are we? I think we're with Eddie.
Eddie, okay.
Eddie? You want to start off with the workspace attributes?
Okay, well, Eddie's getting mic'd up. I'll talk.
Here, you stay mic'd.
Okay. Okay, one of the, if you were to go to the summary page for your workspace, you'll see that there is a list of things called workspace attributes. Now, these are just key value pairs. And so here what we have is attributes that are not associated with a particular entity, but are associated with the workspace itself. And it's here that you might go ahead and provide references to, for example, reference genomes or other files that are going to be used in multiple places in your analysis and are not tied to a specific entity. That's all that I'm going to go ahead and touch on right here, but because we aren't going to be exercising using workspace attributes in the analysis that we're doing today, but that's the purpose of them. And you can go ahead and use this user interface to go ahead and add and change these workspace attribute values. And these workspace attributes can be mentioned in the method configuration and hence can be used as input into the workflows.
So basically, so far we've kind of showed you basically the data. So data is good, but you need to do something with the data. So that's where methods and configs come in. So from a high-level perspective, a method is basically a script, a program, a workflow that encompasses some procedures, some analysis program, scan-a-bam, or whatever it is on your data, okay, from a high-level perspective. So that's what a whittle is. A task is basically a component of a workflow. A task is kind of the fundamental unit of execution. When you run something on Google Cloud, there's basically a 1-1 mapping of tasks and virtual machines.
So what that means is that suppose you have a pipeline like the one that we'll be running for the clustering that has 4 steps in it. Each step is a separate executable. Each step is a task. And that the way that that's going to be running on the Google infrastructure is that we're going to go ahead and parse the workflow that's described in the whittle. It'll identify what all the individual tasks are, determine what the dependencies are, and then go ahead and launch each of those tasks in a virtual machine in the Google Cloud. Now, and some of those tasks, based on the whittle, are going to have to be operated on sequentially. Others can go ahead and operate in parallel, and the system will figure all that out for you. Yeah, okay.
All right, so this is our slide that's basically what we've been talking about. So workflow, as we mentioned, encompasses one or more tasks, the indivisible units of execution. One thing that is important to know about tasks is that a task can be supplied with a runtime environment in the form of a Docker image. So what that lets you do is on your local machine, you can craft what's called a Docker image specifying certain programs or data that should be available in the tasks. And that's a little bit maybe beyond the scope of this workshop to talk about details of that, but it's basically copying your runtime environment locally and sending it to the cloud so you can replicate analyses up there. So here, I want to show this diagram here about the workflow. So here, imagine these 3 boxes being inside another box called workflow. Okay, so this workflow has 3 tasks, step A, B, and C. So what you can see here is that step C is data dependent on step B, and likewise, step B is data dependent on step A, is indicated by the arrows. All right, and so one thing that FireCloud uses to convey this relationship, and not only the relationship, the commands to run that you want to run in your analysis, is the WDL. All right, so WDL stands for Workflow Description Language. It's basically a file that defines a workflow, it defines tasks, it defines the runtime environment of the tasks, which I touched on earlier. It defines the commands that make up a task. For example, samtools index, BWA align, mutect, encrotator, whatever your command is, that is what is in your task. So for example, in this step A, our first task in this little workflow here, we're running tool.a.py from Python. And the Docker I mentioned earlier, so the runtime here is a Docker image as indicated by the Docker keyword here. And the name of the image is correspondingly been named as tool A. And in this case, what's going on is the output is being captured via the standard out. That's what this line here in the output says. So step A sends its output to step B via the standard out. Oh, sorry, sure.
Okay.
I think this.
Yes, we'll make the PowerPoint available. And just to reinforce what Eddie is saying here, suppose you have a workflow consisting of several tools and you want to go ahead and run it on FireCloud. All of the individual tools, the executables, you have to go ahead and package them as they have to be Dockerized. How many people here are familiar with Docker technologies? Okay. Well, this is we're focusing mostly on the analysts. We're going to have a tool developers workshop in a month or so. But basically, each executable unit is packaged as a Docker image, and it resides in a registry of Docker images. And what we describe in WDL is how those individual executables are going to be threaded together to create a workflow. And that's what the workflow description language does. The workflow description language, that WDL file doesn't reside on the Docker registry. It resides in something called the FireCloud methods repository. So when you want to go ahead and run a workflow within FireCloud, you go ahead and reference this workflow description language file that's in the methods repository, and that file contains all the references to the Docker images, and FireCloud will go ahead and retrieve those files from the Docker, from Docker Hub, from the registry, go ahead and instantiate those Docker images in a virtual machine and run that task. And it does it for each of these individual steps. And it handles all the wiring of taking inputs, outputs from one and feeding them into the input of the next one. Okay. All right.
All right. So basically, we mentioned WDLs and workflows and tasks. So far we've only kind of mentioned them sort of in the abstract, kind of independent of the data. So to sort of bridge the data and processing gap, to bring those together, that's where the method configuration comes in. And I think Chet mentioned earlier, and I think some people raised their hands earlier, that there's some familiarity with the Firehose. So the method configurations are analogous to the task configurations in Firehose. So basically, to cause that bridge, a method configuration is created from a method, and where the method specifies inputs and outputs via some variable name, the method configuration allows you to specify which data in your workspace will become those inputs and outputs. Okay, so to get some hands-on experience with this, we are going to import into our workspace 4 method configurations from the method repository. So to do that, we go to the method configurations
Before we do this,
tab.
earlier I had mentioned that the method repository holds the workflow description language, the WDL files for each workflows. It can also go ahead and store method configurations so that go ahead and would allow you to actually go ahead and run that workflow within a particular data model. And so here what we've done is to speed things up, we went ahead and published to the method repository 4 method configurations that will allow us to run 4 workflows. And Eddie's going to walk you through importing those method configurations into your cloned workspace.
Right, and you have the names of those method configurations in your README file if you need them.
That's right. So what I do is I go to my method configuration tab, then I go to import configuration, and then I click on configs only to start subsetting. And then what I do is I can click on workshop here, workshop, and then via that keyword, the 4 method configurations that we want to import become visible, and only those 4. So basically, now we're just going to import them. So to do that, we go to the first one here, then we click on it, and then we say import. So upon importing, we're taken back to the method configuration screen. If we were to want to work on this method configuration, we could start doing things with it now. But for workshop purposes, we have to import some other configs. So we click back on the tab, then we go here, and we basically repeat the process 3 more times.
Yeah. Yes. Nobody can see those method. You can't import them.
Yeah, it looks like it wasn't imported, but then if you click on method configurations, it's there. Yeah. So click on it again, and then you'll see it's there.
Wait, so you, wait.
We're just getting it. I think what happened is when we clicked import, then it gave an error message that said imported. Then if you clicked on method configurations again, it actually did.
Okay, all right. This may be a congestion issue, maybe because we have lots of people doing the same thing at once. It isn't responding in time. And you're probably also all wondering why we have to walk through this step 4 times. Wouldn't it be much nicer if we could just go ahead and identify the 4 method configs and import them all at once? And that's something we'll go ahead and add.
We'll
Oh, no. Okay.
All right, how many people are okay on the protein expression?
Well, what they can do is we can create another expression.
Okay, so that's right. So we'll go ahead and launch our protein expression.
Well,
So
maybe
first,
I'll
I
do
make
that
sure I'm in
during
the
the-
method configurations
I'll do it
area
during
by
the
clicking on here,
break.
and then I want to be
I have a computer.
clear on what I'm clicking on, so I widen my column. And then I go to the protein expression, and I click on that one. And before we launch, I just wanted to briefly.
Actually, why don't you launch first
Oh,
and then
okay.
show.
I'll launch, and then while we wait, we'll tour. Okay, so click on your protein expression, and then go to launch analysis. And then, so notice that it opens up on the sample set because this configuration has set as its root entity type the sample set. So we only have one sample set, so we click on our ACC, and then it's selected as indicated by the yellow highlight, and then we say launch.
So if you had tried to launch this on participant or
Oh, God.
sample, it would have failed.
Oh, my God.
Entity type, just so you know.
I don't know if anyone else saw that.
Oops,
Yeah,
that's not good.
no. We, as,
Try
so,
that again.
so,
That's the protein.
so notice that we have our submission here, so something made it. Something made it.
Look in the submission. That's usually.
So this is submitted, our notation is a few seconds ago.
All right, so now we're in the monitor tab. You should see your launched analysis. You should be able to now go into the monitor tab.
You just go down and you go into that workflow,
Yes.
and it's running.
It's running. Yep.
You can see on this workflow 4 tasks, right there. You can see this is the one that's currently running. So we're holding off on the other.
Yeah,
Yeah.
during the upcoming break, I'll go ahead and see if I can go ahead and fix the problem with the other method config.
Right, so remember how we talked about the workflow picking up of tasks? Now here you're actually seeing the run status of these 4 individual tasks. In addition, you can see error files, input, output files. So if something fails, you can actually go in and you'll get some information.
And since we are in this project, FireCloud BroadE, this job is being charged to the billing account associated with that project.
All right, you want to do the method configs too?
So does this page update when statuses change, or do I need
Yeah, you have to go ahead and click on the tab.
To refresh, yeah.
Right, clicking on any tab will refresh that tab. Okay, so let's take a look at the, I was almost going to talk about this earlier than launch it, but now that it's launched, we can, while we wait, let's look at the method config. So, so let's just take a little tour here. So the first thing we can see is the name, and then which identifies it, and then we see the root entity type is sample set. And consistent with that root entity type that the method config is intended to act upon was that the sample sets were highlighted when we went to launch or immediately selected. So next is
Okay, so I'm going to point something out here now. So the root entity type was a sample set. And you can see that these are the input parameters for the workflow. And one of the input parameters is this identifies the workflow, this identifies the task in the workflow, and this identifies that particular task's input value. You notice here it says this dot protein expression. This refers to the root entity type. The root entity type right here is a sample set. And if you recall, what we did was we added to the sample set an attribute called protein expression, which had protein expression data. And so what this does, this binds or this maps this entity attribute value to the input for this task in this workflow.
Could you use the mouse? It's a little.
I am. Oh, use the mouse. Oh, I see. Okay. Yeah. You can't see that? No. Okay. All right. So once again, on the left side, we have statements that identify all of the inputs to the workflow, identifying what tasks they're associated with. On the right side, we have the attribute values that they're bound to. And we do this both for input values and output values. For the output values, what we see is this is identified in the WDL as one of the outputs for this step, top genes for cluster expression file. That's the top genes for cluster is the step or the task in the workflow. The output is expression file. And what we're doing is indicating that we want to write this back to this new attribute, clustering underscore CNMF underscore RPPA underscore expression file. So once this workflow runs and completes, we can revisit the root entity type, which is a sample set, and we'll see these newly populated attributes. There'll be a whole bunch of new columns in the data model, and it will have these attributes and these values associated with it. So what method configs are, just to reiterate or summarize again, what method configs do is bind the inputs and outputs of a workflow that's defined in the WDL to attributes that are in our data model.
Right, and right now we actually would have had you look at the method configs for the gene expression, because this is actually, we're running the same clustering algorithm on two different data types, and the difference is right here in the method configs. Where it says RPPA, we would have mRNA-seq. Some of the top genes actually gives a number for gene expression. I think it's 1500. So you can begin to understand how you use the method configs to bind the data there.
During the break, I was able to create a new configuration file, and this one you should be able to import into your workspace and launch against the sample set entity. So go to the method repository and then do a search on configs only and then a filter for workshops.
Or new. New.
Actually, probably what you want to do is do this the other way around. I'm sorry. So start with your workspace. That was the exception, it didn't run. That's about, but then we're going to go to the method configurations, and you're going to go import configuration, configs only, and do a search on workshops. And this is the one that you want to go ahead and import. Okay. Go ahead and import it. And then we're going to try to launch it, and I'm going to keep my fingers crossed. See what happens. Ah, there we go. It's running.
Okay.
Okay.
Go to the monitor. Look like
I'm going to go to the monitor tab. And it's running. And we can see, so this is this workflow for clustering involves 4 separate tasks. The first task, so what we're doing is clustering samples based on expression data. In this case, it's gene expression data. And so the very first step that you do when you're clustering based on a gene expression profile is, rather than doing the clustering based on all 22,000 genes or whatever is in your expression file, you're only interested in the top number of genes that show a significant amount of variability across all of the samples. And so the very first step is top genes for clustering, where it takes a look at the top 50, I think we set it to look at the top 1,500 genes in terms of variability and expression across the samples. And we see that it's running right now. Once those top 1,500 genes are selected, then we're actually going to go ahead and run the clustering. The clustering algorithm we use is called consensus non-negative matrix factorization. It's been shown to do a more effective job than hierarchical clustering or other clustering algorithms on expression data. If you're interested, after the workshop, I can show you on the whiteboard how the clustering is done in terms of how you do how it's done. It has some similarity to principal component analysis. After that clustering is done, what we do is we go ahead and do the clustering for 8 different numbers of clusters. We try to do the clustering for k equals 2 through 8. After we've done that clustering for k 2 through 8, we want to choose the one that is the best clustering in terms of doing the best job in grouping the samples together and minimizing the differences between in the profiles between the members of each cluster. And that's done through this third task, which is called CNMF select cluster. And it'll go ahead and choose one clustering, whether it'll be for 2 clusters, 3 clusters, or 8 clusters, to choose which number of clusters, and that'll be the result of the workflow. And then what will happen is we'll generate a report for that, which will show all of the clustering information. And in addition to the report, it's also going to generate a clusters file, which for each of the samples, it'll indicate which particular cluster it's in.
And if you recall, those were specified in the method config. So if you looked at, let's see, we looked at the protein expression, but the number 8K was specified there.
We can go ahead and take a look at that right now.
Right.
Yeah,
Look at the one in the...
So if we go to the method configuration, and we're looking at the new one. And the input values. So this is for clustering of gene expression data. We're only interested in the 1,500 top clusters. Only the 1,500 top genes that show the greatest variability. The K-init, the number of clusters, is going to range from 2 to 8. The report that we're generating, it's a RNA-seq report. And the input is going to be the gene expression file, which we already uploaded into the workspace as metadata or an attribute associated with the sample set. The statistical measure that we use for comparing the different clusterings and see which one is best is the Pearson metric. And that's it. And then we go ahead, and these are all the outputs that are generated. We have a bunch of plots that are generated, and we have the most important thing that's output is these membership files. Okay. So right now, let's go back and do one more peek at.
Look at the monitor.
Yeah, I'm going to go to the monitor right now and see where we're at.
We have a question from.
Oh. Yes.
Why is the top number of genes, why is that a string instead of an integer?
It's a great question. The way that the method configurations work, there are only 2 types of inputs. They are strings or files, but the WDL explicitly defines whether the input is a string or an integer or a file, and it'll go ahead and convert that string to an integer.
But the other parameter was set as an integer. I was wondering.
In the WDL. I don't think it was it set as an integer in the method configuration?
Yes.
Okay, let me take a look and see.
So the first variable is
int.
a string, but the second variable is an integer.
That's a good point. That seems to be how it was defined in the WDL, in the workflow description language. It may be the tool itself. Actually, my guess is that what's happening is you have 2 steps. This is for the NMFS consensus clustering, and this is for selecting the top genes. And most likely, the tool for the top genes, that input value, it assumes all the inputs are strings, and the conversion to an integer is done internally within the tool. But it looks like this particular step assumes that the input is, in fact, going to be an integer. When you create these method configurations, the way that they're created is you go ahead and import the WDL into the workspace. That import doesn't actually move the WDL into the workspace. What it does is it creates a template for the method configuration where it lists all of the inputs and the outputs, And then these values right here are left as blank expressions, and you have to go ahead and fill them in. So whether or not an input is cast as a string or an integer or a file, that is determined by what's in the workflow description language for that tool. Okay? All right. All right, so we'll wait a little while and see what happens
Go
there.
to the monitor, it looked like one of the jobs failed.
Oh.
Let's just check.
Just check to see if a job failed? Okay. Wait, one of these?
No, okay, and it's
No.
done. Okay.
One is done, yeah, so we can go ahead and take a look at that. So this is the clustering that,
It's bad kind of done.
yeah.
Not the good
Okay,
kind.
Not the good kind of done. So, well, this is a good exercise. You didn't plan it that way, but invariably in the course of using this system, you're going to run into errors, and it's a good way to sort of see how we go ahead and try to debug what's going on. So we know that the order of the tasks that occurred was the first thing we're going ahead and looking at the top genes for cluster, and that seemed to fail. You can always go ahead and look at standard error. Oh, that's interesting. I'm signed out.
Oh, did yours work? Ah,
Yeah, most of it.
oh, everyone else's worked.
Okay.
Oh, yours failed, okay. Well, at least we're not alone in this. I wonder if this was because we had 50 people kicking it off all at once.
It very well could have
How
been.
many people finished? Runs finished? Okay, excellent. This is good.
Yeah, okay, so probably what happened was you guys are just faster than us, and you got into the queue before we did.
Yeah,
So-
go to the C burger and let's just open up the results so
Okay.
they can see how to look at results.
Okay, I'm going to do that through a,
Oh, no, you can, it's on my account.
oh, it is? Oh, I
Yeah.
shared it with you.
Yeah.
Okay. Okay. You have to kind of appreciate that when we run these workshops, they really stress the system because we essentially have 50 people doing the exact same operation over in the same 4 or 5 minute interval. And in real life, that's not going to happen.
We hope.
Well, okay, so
No,
it's Seaburger?
yeah. All right, so remember how Julia Childs used to pre-cook things?
Yeah, this
Okay, we
is,
pre-cooked it for you.
this is our turkey.
Oh,
right?
okay, so, sorry, hold on, Chet, we got some questions. I'm sorry?
Is there an account-specific concurrency limit or something? Why is there a specific queue dedicated to BroadE?
No, I don't want to go assuming that failed because of that. We should look and see what the reason is for the failure, because I don't think that actually was the case, given
Okay.
that everybody was able to go through no problem. It's probably just that one of the inputs in the TSV was wrong. It could have been any number of things. So it's a good thing to possibly look at if we wanted to show how you can see where errors are coming from.
Right, that might have been the case. Maybe we made a,
Limitation.
yeah, that's a
Yeah,
good point.
Could
too.
be. We'll double check that. But anyways, we pre-cooked it for you.
In terms of, there's limits that are set on the projects that you run within, so there's a certain amount of, basically.
No, just go to the data mine.
Set up.
Okay.
In the back end that's being used, things are actually retry when that
And then you're
gets,
just gonna filter
when
to
that
the
gets
recording.
reached. So
But
it
hold on,
changes
do it with
the
that.
time at
Wait till, wait.
all the stuff within our system in terms of currency and limits we're still working on.
This one,
That's not
RPP, RNAC.
Yeah, those two.
sort of inherent within the system. If anything, there may be some queuing like Firehose has, where you may wait for somebody else's jobs to complete before yours can. There's some sort of inherent limits that are other than query limits, which you can often like to. If you go very high with your limits, the chances of you exceeding a lot of money spent in one day.
Were there other questions?
So I think the question was a few moments ago, and maybe still is, trying to look at the standard out, or look at an output from the run, whether it failed or.
And what we're going to do, yeah, first what we're going to do is show you the turkey that didn't get burnt.
Okay.
Okay, so this is the one that Julia Childs did earlier on, and it worked fine. And then we can go ahead and see where I messed up the
I have a
recipe.
question about viewing output, but I'll ask that.
Yeah, and we'll go ahead and show those. So As you can see, what's happening now is this is actually within the data model where we've run all of the various workflows that we want to do during the course of this workshop. And you can see that we've added a lot of new metadata columns or a lot of new attributes. We're interested right now to take a look at the outputs that are associated with the clustering algorithm. And so what we're going to do is I'm going to go ahead and I'm going to turn off, I'm going to filter away all of the entities, all of the attributes, and then I'm just interested in the entity name, and I want to see the report for the protein clustering, and I want to see the report for the gene expression clustering. And here they are right here. We can go ahead and open it. Now this is something called a nozzle report. Those of you who use FireCloud or are familiar with FireBrowse are going to be familiar with these reports. What we did was RPPA is protein expression data, and we can go ahead and take a look at the results for this. And here we can go ahead and see the clustering where the best clustering for the protein expression data was based on 2 clusters. And you can go ahead and sort of see the results. This is the clustering that was done through the use of the NMF clustering. This is a clustering using standard hierarchical clustering, and you can see that it's not quite as effective. You can also go ahead and look at this information on the methods that were used, and you can drill down in the silhouette widths.
Well, marker proteins, too.
Oh, marker proteins, okay. So these are the proteins that drove the clustering. Yes?
My question I had a few moments ago was, so to access that, you took down the output, you got the files details open up, then it was file size, open, regular fit out.
Yes.
For me, and I think a few other folks, when we click open, it brought up a source FireCloud status error on every single possible output. So I was wondering if, but I was able to find outputs by going into the school bucket. Which is that your method is opening here.
Yeah. Well, this is on a analysis that was run last night in a separate workspace.
So that might be a symptom of.
Yeah, we seem to have a permission thing somewhere here.
That's great.
Okay. We'll sort out. But what we're doing is accessing the outputs based on the data that was written back to the data model. You can also go ahead, and if you were to go ahead and go into the monitor screen, and this is, you can see that we've submitted a number of analyses. Let's take a look at the one that was submitted last night. And this is the single, actually this is the wrong one. Okay, so I'm interested in the clustering analysis, in the clustering workflow, and we were looking at protein expression data. This is the workflow. And these are the 4 steps. And the final step is the report generation. And if you were to go in here, you could take a look at what the inputs and the outputs were for that step. And you can access that same report by looking at the task level outputs. So if I were to go ahead and click on this, it's the same report, and I can go ahead and open it. Okay? Yes?
So just to be clear, this beautiful report is just, you've got a program that generates some HTML.
Yes. This is.
It's not part of.
It's not part of FireCloud. FireCloud is a platform for running the workflows, and quite often what you want to do is design your workflows so the final step does report generation. It's all about the content and the executables that we're adding to FireCloud.
And all the other output files are there in that list that we just showed you. The nozzle reports are just a nice way to roll them up.
Yeah. Okay. All right, so this is, okay, where are we now? Let me go back to the, okay, so we were going to do a quick view. So some of you were able to go ahead and successfully run the protein expression, And were you able to go ahead and look at the resulting nozzle reports? Okay, good. All right. This slide just describes what the clustering does, the features of the clustering, and once again, I can describe it in greater detail after the workshop for those who are interested. Okay, now we're going to, all right, so hands-on aggregate analysis. The next step is to go ahead and what we have, let's go back and take a look at the outputs for these two types of clustering, where we clustered based on protein expression and we clustered based on gene expression. Let's go take a look at the data. I'm going to go to sample set. Oops. Okay. Now what I'm interested in is the results of the clustering. And this is the best cluster files. And there should be one for mRNA-seq and one for protein expression. Okay, first thing to notice is this is an output file. And so the output is written to the bucket that's associated with the workspace. We're going to go ahead and open it. And so what you have here is this goes ahead and it chooses it indicates what the clustering is. So this column right here are the identifiers for the various samples in the sample set, and this is their cluster assignment. And so these are in cluster 1 and these are in cluster 2. All right. There's going to be a similar file for the gene clustering. And for the gene clustering, though, the best clustering was using k is equal to 3, breaking it down into 3 distinct clusters. And here, once again, you have the cluster assignments for each of the samples. All right. And you might ask, well, there are a lot more samples for the gene expression than there were for the protein expression, and that's because for this particular sample set, for the ACC cohort, we had gene expression data for all of the samples, but we only had protein expression data for a subset of the samples. The next step is what we want to do is to go ahead and it's a very simple action. All that we want to do is take these cluster assignments and combine them into a single file. So we have a list of all the samples, and we indicate what their clustering is for protein expression, and in the same one we indicate what their clustering is for gene expression. So all it's really doing is just concatenating columns together. That's what this task does, and we're going to go ahead and run it. There's a method configuration for doing that. Here it is, aggregate molecular subtypes. And this is actually quite general. It's part of the GDAC pipeline, and in the GDAC pipeline, you can do clustering across all sorts of different types of data types. To keep things simple here, we just chose 2 particular data types, protein expression and gene expression. And all that we did then is provide as input these 2, these are all optional inputs. The 2 that we specified was the clustering results for the protein expression, which is right here, and then we had the clustering results for the gene expression, which is right over here. So we're going to go ahead and launch this analysis against this workspace. And there it is. So those of you who were able to successfully run the gene expression and the protein expression should be able to go ahead and do this.
Okay.
Okay.
This piece doesn't take long to run at all. I think it's like 10 minutes or less. All right, so we wanted to talk about TCGA controlled access and dbGaP authorization because presumably a lot of you are interested in accessing or using this data. So FireCloud users can co-analyze and compute on TCGA data, both open or controlled access. Controlled access data is de-identified and it may be unique to individuals. TCGA is really, really sensitive about proper use of TCGA data, and they will shut labs or groups down if they feel there are data infractions. It's actually really complicated. For example, somatic mutation MAF files are considered public. Germline are considered, open access. Germline are considered controlled access. And then it gets more complicated if there are somatic mutations on the edge of the exons, they can be controlled access. So it gets messy pretty fast, and it's constantly evolving, and they sort of change what they say is controlled and open sort of regularly. Essentially, the technology is ahead of the ethical thinking around TCGA data. As all of you who've worked with genomic data before, you know you actually can identify things in data, even data that's at a pretty coarse granularity. So controlled access data is specifically things like BAM files for DNA or RNA-seq, somatic germline mutation calls, SNP array and birdseed files. FireCloud users with dbGaP authorization can access controlled access data, and we'll sort of walk you through what you need to be able to do in order to do that. And basically, the way that's done is secure authentication through ERA Commons. Open access data, as you saw, is public and it's de-identified. There you see clinical data. You can go to the TCGA data portal and read all about the data itself if you have any questions. And all FireCloud users have access to the open access data. It's in your workspaces right now. Oh, we actually already looked at that. Okay. Open access workspaces will be reader level access. I think we've been through this. Controlled access data workspaces will be limited to dbGaP authorized users who have reader level access. Sorry. Importantly, FireCloud users are responsible for sharing controlled access data properly. So if you're working with controlled access data, really be mindful how you share it. And what we've done is we've actually built some additional security in that helps you contain the controlled access data. And Chet will get to that in a moment. So I'm just going to walk you through exactly what you need to do. I've already had a couple of questions for people. Number one, you must have an ERA Commons account. How many people in this room have an ERA Commons ID? Yeah, okay, a fair amount. You must have dbGaP authorization for TCGA data. So dbGaP authorization is through a PI. So if your PI has approval, then you can get authorization. You need to be sort of one of two things, a downloader or a collaborator. So you actually must be on the dbGaP application. Once you're on the dbGaP application, then they have a whitelist for all dbGaP authorized people, and our system hits that whitelist regularly. And then this is sort of a random small thing, but it's true. You must have logged into dbGaP at least once with your ERA Commons ID. So ERA Commons accounts, your organization must be registered with ERA Commons. If any of you want to access the data, go to your PI, ask for an ERA Commons ID. They're easy to be set up. ERA Commons IDs are actually done at the organizational level. And it requires a signing official from your institution. So there's more, there are links here that can give you more information. Okay, so Anyone who wants to have dbGaP authorization, you must submit an application through dbGaP authorized access. It's sort of your standard application. It's really not bad. We're happy to share our successful applications with the text if anyone wants to see that. It's sort of your standard application. You explain what you're trying to do, what you want to do. It's a web application, but it's not that bad. Your PI, all applications go through your PI, and then lab members are covered but must have their names specified again, and your signing official. Oh, I already said all this. Okay, moving on. And there's a link at the bottom there for the application process if anyone has questions. Okay, so how does this happen? How does this happen within FireCloud? If you look at your profile at the top corner of your page, let me walk to mine. Here's my profile page, and scroll down to the bottom. Here's linked to my NIH account, and you can see I'm an authorized dbGaP user. And this link here can take you to your ERA Commons application. Okay, so you'd need to log into FireCloud, click on your user profile, and then click on your login to NIH. If any of you have your ERA Commons, you could actually do this now. Log in, see if you can get in. This takes you directly to the ERA Commons front page. Once you put in your username and password, it will take you directly back into FireCloud. And if it hits approval there, it'll take you directly back. This is the dbGaP front page. You should be able to log in because your ERA Commons ID also gets you into dbGaP. If you want to know whether you actually have access granted, you can actually go on. You should see available TCGA project here, and you should actually be able to see that access has been granted. So just to summarize this, you must be able to successfully link to ERA Commons, and you must have dbGaP approval for TCGA controlled access data, and then you'll be authorized. I have a note here that says it may take up to 24 hours for FireCloud to recognize you, but the reality is it's much shorter than that. I don't know what it is now, a few hours? Yeah, at most. So that is pretty quick. Okay, and then one other thing. For security reasons, you're asked to relink your account regularly. So periodically, I think it's a week.
The way it's set up is that if you go ahead and link your FireCloud account to your ERA Commons account, and you go ahead and launch in and say that you go ahead and clone one of the controlled access workspaces, and then you launch an analysis, and you're doing a lot of work, and you know that analysis is going to take 8 days, a week, or something like that. If you don't revisit FireCloud in that time, your analysis is going to go ahead and run. The linkage between your FireCloud account and your ERA Commons account, when you first establish that linkage, is good for a month. But if you go ahead and revisit and log back into FireCloud in less than a month, it'll go ahead and request, if it's been 24 hours since you first established that linkage, it'll ask you to go ahead and relink your ERA Commons in your FireCloud account within 24 hours. And the reason for doing it this way was the NCI initially wanted this linkage to only be good for 24 hours. But we told them that people are going to be running analyses that might take days, and we don't want their linkage to expire during the course of running this analysis. And so this was the compromise that we put in place. So if you go in, run your analysis, don't go back and visit FireCloud during the length of that analysis. As long as it takes less than a month, you're going to be fine. As soon as you go back into FireCloud, if it's been more than 24 hours since your account linked, you're going to have to relink it. Okay?
Hi, I think you're up on the...
Okay. All right.
Though we might want to have people kick off the last analysis.
Okay, have they done that yet or not?
No, we one more, the clinical correlation.
All right, well, let's go back and see if we have successfully completed the this is the one we're working in now, right, Megan?
Yep.
Okay. Just went ahead and ran this. And it's done. So this was the analysis that actually ran the aggregation. And you can see that it was just a single task. It was going ahead and aggregating the clusters. And I'm going to actually take a look at the output directly from here. It goes ahead and created a merge cluster file. And here you can see that, so these are all the samples in, okay. These are all the samples that are in that cohort, and we have two clusterings, one protein, one based on this is based on protein expression, this is based on gene expression, and here we have the cluster assignments for each of them. So all that we did was go ahead and merge these columns together. And the reason why we had to do this is because the next downstream workflow, which is to actually run a correlation analysis to correlate these clusters, which potentially represent different cancer subtypes associated with the cohort. We want to be able to correlate them with clinical parameters. And that's the workflow that we're going to go ahead and launch right now. I should note that this combined workflow also appears it was written back to the data model, and I believe that we can quickly search for it. It's aggregate molecular. I'm looking for the output. Is it this one? Nope, it's not it. Maybe that is it, protein expression. Maybe that is it. Yeah, that is it. Okay, so it was written back to the data model, so now it can be used as input for the next flow, for the next workflow. Let's go to the monitor screen. Method configs, and which of you, how many of you were able to successfully run this aggregate task? One.
I succeeded, but I don't see the third column. Yeah, it's just the first two columns.
The third column in the output. When in the data set, when you were doing what? When you were
Did both the gene expression and the protein expression?
Yeah, the gene expression took a long time.
Yes.
Yeah, okay. Yeah, it may have not been, both files may not have been
It may
done.
not have completed at that time.
Yeah.
Oh, okay.
Yeah, so your gene expression, which I guess we just launched.
Yeah, we launched that afterwards.
Yeah. Okay. So now what I'm going to do is launch, granted a lot of you aren't able to do this because you don't have that fully aggregated set of clusters. For the purposes of demonstration, I'm just going to go ahead and launch this final workflow, which does the correlation between the clinical parameters and the clustering from the protein expression and the gene expression.
So a question about these sort of issues.
Yes.
Is there any notification other than keep going and checking the website to, like, when you have this sort of thing?
You mean email?
Yeah.
Not yet.
Not yet. And
Eventually there will be.
yeah. And the way that if I was an analyst doing this, probably what I would do is rather than having 4 different workflows, I would have constructed a single workflow that did all of these steps. And that single workflow would make sure that each kind of downstream task wasn't actually run until all the upstream results were in place. And if there was a failure, it would go ahead and report that for the entire workflow. But we wanted to break it up into pieces, which is why we were writing things back to the data model. Okay. Just going to go ahead and launch this. Okay. And we'll see what happens. Okay, while this is running, let's talk now a little bit about derived data. So we know that there are two categories of TCGA data, open access and controlled access. The NCI also stated that they view all data that's derived from controlled access data to also be controlled access. So if you run some kind of analysis on BAMs in the TCGA data set, and those BAMs are controlled access, and the results are also going to be viewed as controlled access. And they wanted us to provide a mechanism of ensuring that the only people who can access that controlled access data are those who are dbGaP authorized. You can envision a scenario where I'm dbGaP authorized, I go ahead and clone the BRCA cohort, controlled access workspace. I go ahead and run some kind of analysis on it, and I share it with a collaborator. And that collaborator is not dbGaP authorized. I can still share that workspace with the collaborator. And while the collaborator, because they have access to that workspace, they still aren't going to be able to get to the original BAMs, because those are in a bucket that only people who are dbGaP authorized can access. The results of the analysis that I ran in my clone are going to be written back to the bucket that's linked to the workspace. And if I choose to share that with somebody who's not dbGaP authorized, they're going to be able to access that derived data, which NCI views as controlled access. They wanted us to prevent that from happening. And so what we have done is we ended up addressing this at the workspace level. So when you create a workspace, you have to indicate whether or not it's going to contain controlled access data. Just the same way when you create a workspace, you have to specify what Google project is linked to it. You also have to say whether or not it is going to contain controlled access data. Now, if you are cloning a workspace that already does have controlled access data, you have no choice. The clone also has controlled access. If you clone a workspace that doesn't have controlled access data, but you know that you are going to be putting controlled access data in there, or if you go ahead and create a new workspace and you intend to go ahead and have it have controlled access, in those cases, you have to go ahead and indicate that it's going to be controlled access. So suppose I go ahead, clone BRCA. My clone is going to be also a controlled access workspace, and I go ahead and explicitly share it with a colleague who is not dbGaP authorized. Because I explicitly shared it with that colleague, when that colleague goes into FireCloud and looks at the listing of workspaces, they are in fact going to be able to see in that workspace listing the workspace that I explicitly shared with that person. But if they were then going to go ahead and go into that workspace, they would be blocked. And a message would come up and say, sorry, even though you have access to this workspace, we can't let you into it until you've demonstrated that you're dbGaP authorized. And that could occur for a couple of reasons. That one may be that my colleague is dbGaP authorized, but the colleague hasn't linked the accounts. Or maybe they had linked their accounts and the linkage had expired. That'll be a signal to the colleague to say, you know, if I really want to look at this data, I better go ahead and link my accounts. And I'm going to walk you through some of those operations right now. So the bottom line is that we go ahead and control access to derived data that's viewed as controlled access data. We do at the level of granularity at the workspace, not the file. It's easier to do it that way.
Right, and this system of protecting data is actually extensible. Right now, it's only in use for TCGA data, but eventually it could also be applied to other data sets that may or may not have controlled access. Or, for example, you could imagine a lab sharing and having these kinds of security controls on it. So it's extensible, and eventually we'll have other applications.
Okay, so here we're taking a look at the two BroadE workspaces in the system. One is open access, one is closed access. Suppose I want to go ahead and clone this closed access BroadE workspace. I go ahead and click clone, and this is Hannah clone. It says that the cloned workspace is going to automatically be protected because this workspace is protected. That means it's already going to be dbGaP. It's a controlled access workspace. I'm going to go ahead and do that. Now, I probably made a mistake by choosing the BroadE workspace because that has about 2,000 entities. It's one of the largest cohorts, but it went ahead and cloned satisfactorily. And you can see that in the panel it says this is restricted workspace for TCJ controlled access data. Now, Megan can go ahead, and if she wants to, she can go ahead and share it with me. And give me read access to it. Okay. Now, in a little bit, we'll go ahead and I'll log in as myself. I am dbGaP authorized, but I had let my linkage expire, so I shouldn't be able to go ahead and I should be able to see a listing of this workspace, but I won't be able to actually enter it, and I'll go ahead and demo that for you. The other thing I wanted to show you is, so we go back to the workspace listings. BRCA. We have the open access one. Okay, here we actually, this is great because we can see this is the one that we cloned, and you can see that it's also tagged as being restricted. Let's go ahead and clone this one. Or rather than cloning it, let's go ahead and create a new workspace. I'm just going to create a new workspace. And you can see now that I have the option of indicating whether or not it's going to be a controlled access workspace. I'm going to say that I do want it to be a controlled access workspace. And this is just a workshop example. And there it is, and it's restricted. Okay. When you have so you can, when you clone a workspace that is, just to reiterate, you clone a workspace that's controlled access. The clone has also controlled access. You clone a workspace that's open access. You have the option of whether or not you want to say it's controlled access. If you create a new workspace, you have the option of saying whether or not it's controlled access. If you have a controlled access workspace, you're going to be able to copy entities from a controlled access workspace into that new workspace you created. If you have a workspace that is not controlled access, if you try to copy entities from a controlled access workspace, any entities, regardless of whether or not they contain controlled access data, because they're in a controlled access workspace, you'll be blocked from copying entities into your workspace that's not controlled access. All right. So all that is that clear to people? Yeah, all right. Okay, let's just do a quick, let me bring up, I'm going to log in under my account. Okay. Restricted. Where was the one that I shared? Okay. That's interesting. Did I successfully share that, Megan?
I thought you did.
Yeah. Oh, but maybe I didn't name, I named it something else.
Oh, you did.
That's the problem.
You named it example.
Example.
Test.
Yeah,
Yeah.
okay. Example?
Test.
Now let's go back.
What did he name it?
What did I name it, guys? Yeah. But it was with the, yeah, I don't remember. Workshop example?
There it is.
Okay, but let me make sure that I did share it with, oh, I thought I shared it. I guess I didn't share it.
Yeah.
So I'm going to go ahead and share it with myself. Or I'm going to give
Share with the group.
no, not right now.
But actually you can, and that's something, when we make something public, you basically add it to a group. So for organizing labs or specific groups or projects, that's a useful feature.
But right now, I don't think we actually have that ability.
We don't have a group of all these people here.
No, no. Okay, so I'm sharing this with Megan is sharing this to me. She's giving me read access. This is a controlled access workspace. Okay. Let's go back and filter based on workspaces. What's going on here?
You've got something in your filter.
Yeah, right. Okay, so I think we said it was example. No?
Workshop.
Workshop. There it is. No, where is it? What's going on? BroadE. Oh, I didn't save it. Okay. Please let it be there. It isn't still. Burger. Workshop example.
Are you sure that's the same user you're logged in with?
Well, right here I'm logged in. I should be logged in as Chet Berger. Yeah, it's the same. It's the right account.
Chet, it may be because your link expired for controlled access data.
Oh, of course.
No, but what should happen is that I should be able to see it in the listing. But then what I wanted to demonstrate was I see it in the listing that the workspace has been explicitly shared with me, but I should be blocked from actually entering it. And I'm not quite sure why that is not functioning. I don't know. Okay. Well, we'll have to work on that. The way that it's intended, I apologize. The way it's intended to operate is that users are who have, say, who aren't dbGaP authorized or who have let their linkage expire, they should be able to view workspaces that were explicitly shared with them in their listing, but they shouldn't be able to enter them. For some reason, we can't actually view them in the listing right now. All right, so now we're going to go back to the, we ran a workflow for correlating clinical parameters to the subtypes that were identified in the clustering. And we're going to go ahead and take a look at the results from that. Workshop example. I want to go to the other workspace. Okay, this is the analysis that we just ran. It's done. It succeeded. We go into the workflow. You can see that it had two tasks associated with it. One is to actually do the correlation analysis, and the second is to go ahead and generate a report. The report is available either through looking at the task level output. We can view it here, or we can also go ahead and take a look at the report, because it's the link to the report should be captured in the data model itself. So let's go ahead and find it. We're going to go ahead and look at the entity, and here it is, clustering. Now correlate. This is the nozzle report right here. I'm going to open it up. Okay. And this is the nozzle report. So we kind of ran our analysis using two different clustering approaches, or two different clustering on protein expression, clustering on gene expression data. We found that the protein expression data, we found significant correlations with a single parameter, with a single clinical parameter, and that was time to death. For the clustering of the expression data, we found that there were significant correlations with a whole collection of clinical parameters. We can actually view those results by going ahead, and here, these are the results. If they're in red, it indicates that we did have significant results statistically, and we can go ahead and drill down into them, and we can see what the p-values and the q-values were. Once again, this report generation, this is all a function of the tool. It isn't a function of FireCloud itself. FireCloud is the platform that allows you to run these tools. But we did want to show you how, once you've run the analysis, if you have tools that create HTML reports, you can view them directly within FireCloud. Yes?
So I'm looking at my, what do you call the bucket browser?
Yes.
As this is running, and I'm seeing a whole bunch of hexadecimal directories in there.
Okay, those directories are not hexadecimal directories. Well, maybe they are, but they are directories. First of all, remember that the bucket, actually, understood, but they appear to be directories, and those long numbers are reference, I think, job submissions and workflow submissions. So it's a way of organizing and uniquely sort of identifying the results of a particular workflow and a particular task in a workflow.
So if I want to have my data in some kind of intuitive hierarchical format, what do I do?
The intention is to organize your data in the workspace model.
So basically, I go find one of something that's been run and look, or I look in my data tab.
You would look in your data tab, right? The intent, the organization of the files in the workspace bucket, that is primarily intended as it's the storage for that FireCloud uses for tracking the outputs of workflows in a particular workspace. And it's all encoded with those long, crazy names, so it can go ahead and identify for a particular workflow that has a unique ID, the particular sort of task, that job that ran, and the outputs associated with that. Okay. All right. Where are we now? Okay. We're done?
We just did that.
We just did that, right. Well, I did it here. We wanted people to do it hands-on, but people have to wait until, I don't know, you really couldn't do this until you first successfully completed the aggregation workflow, which went ahead and took those clustering columns and merged them into a single file, wrote that file back to the data model, and then you could go ahead and run your correlation analysis. And I don't know how many people got to the point where they could go ahead and do that. Okay. So the next thing is, if you recall, at the start of the workshop, we ran our mini mutation calling tutorial. And I think what we want to do now is revisit that, because I think the tutorial takes an hour or so to run. We wanted to look at the results.
We can look at the results to see if they're
Okay, but we need to go back to the workspace in which they were run. And that was, well, I don't know what workspace you ran them in. You went ahead and cloned them, so what workspace did you run it in?
Search for JN.
JN.
This is the second one.
JN, this one right here, mini mutation column? Okay.
And then the data tab, you should be able to see.
Okay, let's see, make sure everything ran. It was successful. We ran it against both pairs, and they both succeeded. I'm going to, so you want to go to the data tab? And what are we interested in looking at? The HTML? Okay.
Yep, and I think the Onco VCA, but that's all right.
Okay. This
Right,
was,
so
yeah, I'll let you speak to
That's
this.
the results of Onco. Remember the mini, this is a mini BAM. It's got 100 genes, so there's only one column. But there you have it.
So this is a tutorial workspace we're making available to all FireCloud users. So anyone could go into FireCloud and find the mini mutation column tutorial workspace, clone this workspace, got this analysis. It's just to show you, basically how you can run analysis in 30 minutes or so on the mini BAM to get some results.
Okay. Okay, and we took a look already at the view of the, well, I demoed the result of running that correlation exercise. And those of you who were able to successfully run it can also go ahead and view the results. Okay, I'm going to.
Yeah, so here's what we've covered. I think hopefully you guys all know the basics. I'm sure in this crowd there are a considerable number of tool developers and people that really want to be running this from the command line. And we have a lot of those tools. In the next, our next section will talk about how those tools are structured and what they are and how you can access them. But that'll be the end of sort of the hands-on part. But hopefully what we've given you is sort of basic functionality of the UI, how to run and launch analyses. Also, you have some sense of what the what FireCloud holds in terms of workflows, tools, data. So we wanted to just give you an overview. So from here, hopefully this is the list of the things you sort of understand and have done. And from here, we'll sort of just take this next last piece of time and talk about the tool development piece.
Okay, so before.
Any questions first?
Yes.
No, okay.
Okay. All right. All right. So as we said, the FireCloud supports multiple kind of communities of users. There are the analysts who we envision primarily using the graphical user interface for working with FireCloud. But the way FireCloud has been designed is that the entire front-end portal is built upon a public RESTful API. And that essentially means that people can go ahead and, rather than using the graphical user interface, they can go ahead and run scripts that do all of these actions. And those scripts, there are a variety of different ways of writing scripts. You can go ahead and write your scripts, and so they directly make calls to the RESTful APIs, for those of you who are familiar with that. And what we do is we go ahead and provide a online documentation for the RESTful API, and that can be accessed through going to api.firecloud.org. and you all should be able to go ahead and access this. And this kind of shows what the, for example, if you have entities and you want to, these are the examples of the APIs that one would go ahead and call, the RESTful APIs that one would go ahead and call in a scripted interface for this. Another set of tools that's under development is something called FiZ, FC. For those of you who are familiar with FireBrowse, you know that there is a FiZ, and I forget what the acronym stands for, but there is a FiZ command line interface for working for programmatically against FireBrowse. We're providing the same thing for FireCloud. FizFC is written in Python. You can either go ahead and run it as a straight Python application, or if you want, it provides a command line interface, or if you want, you can go ahead and it's also a set of Python packages, and you can import them into your own Python programs and write Python code for interfacing with FireCloud. Yes?
Is that a different version of this?
Yeah, it's different. So you're familiar with Fiz. It's different because the Fiz for FireBrowse goes to, I believe, the GDAC version of FireBrowse, of Firehose, where FizFC is linked to FireCloud's public APIs.
Okay. Does it still store your password in plain text?
No, it doesn't. Though because here what we're doing is using, you have to get a Google. You're going to use the G Cloud, the Google tools for getting an authentication token, and you go ahead and provide it that authentication token. Okay. Okay, so we discussed this a little bit earlier about, well, we touched on it briefly. We are going to have a separate workshop for tool developers, but we wanted to kind of outline the basic steps that a tool developer would use in order to get a tool into FireCloud. The first thing we said was that all tools, these are executables, they all have to be packaged to go ahead and run in a Docker container. You go ahead and do, and I'm not going to go through that technology right now, but the final endpoint of that is obtaining something called the Docker image, which you can go ahead and place in a Docker repository called Docker Hub. You can go ahead and either have tools or images that you put in Docker Hub and they're readable by all, or you can go ahead and have private tools that you write into the Docker Hub registry. If you use private tools and you want to make those tools then available in FireCloud, the only individuals who are going to be able to access those tools are those who are explicitly granted access to it. I think that's the way it works. Once you have your tools in the Docker Hub, then you want to create workflows, and those workflows are going to consist of a series of individual tools that are threaded together into a workflow, and that threading is implemented within this workflow description language called WDL. So the next thing you're going to do is after your tools are up in Docker Hub, you go ahead and define a workflow description language file that defines what those, that essentially takes those individual tasks, wraps them in WDL, and then goes ahead and shows how they're all threaded together. Once you've authored that WDL, you want to go ahead and test it probably before you go ahead and move it into the method repository. One thing that the workflow execution service in FireCloud is a service called Cromwell. And it's this service, or this program, which goes ahead and takes the WDL It goes ahead and parses the WDL, figures out what the tasks are, and figures out the order in which to run them, and goes ahead and launches them. And within FireCloud, it goes ahead and launches them within on the Google Cloud infrastructure. If you want, you can also go ahead and install locally on your local development system a local version of Cromwell, and you can go ahead and run your WDL against that local version of Cromwell just for the purposes of debugging, to making sure that there are no errors in your WDL, to verify that the workflow works. Once that's done, then you would go ahead and publish that WDL workflow to the FireCloud's method repository, which is called Agora. There is a separate command line interface called FireCloud CLI, which is accessible through GitHub, which we would go ahead and sort of show FireCloud tool developers how to use. They would use that command line interface for publishing tools into the method repository in FireCloud. And then they could go ahead and run. We will make this process more streamlined, but this is the process that we currently have for adding tools to FireCloud, but we intend to go ahead and make it streamlined over the coming months.
I don't know if anyone remembers the early days of Firehose. It was really challenging to get a tool in. And Gaddy decided that that was really too hard and you need to be able to get a tool in 15 minutes. So we're not at the 15 minutes yet here.
No.
But we will begin building infrastructure to make all of these steps streamlined, because we know this is a substantial piece of work right now.
Okay, I want to skip going through Docker basics right now and Docker terms. I talked about the FireCloud CLI. You could run this in a couple of different ways. One is to go ahead and install the CLI directly on your laptop, on your development machine, and run it. Or if you want, you can go ahead and we make available a Docker image that has that CLI running in it, and you can go ahead and run the CLI within that Docker container. I talked about Cromwell. So Cromwell is the workflow execution system or service, and it's a program that you can go ahead and run, a Java program you could go ahead and run locally on your system. And you can run it locally on the system and then have it go ahead and run your test WDL, and I'll go ahead and parse it and make sure that there are no errors in that WDL. I talked about FizzFC, which is a sort of a follow-on to the Firehose service selector, which is used for the used in the FireCloud in the Firehose GDAC. We have gone ahead and sort of added a similar utility for FireCloud, and it's currently under development. Okay. Now we have FireCloud resources. I think
Right, so please send all questions through the forum if you monitor that. We have also, a lot of questions can be answered from the user guide. You can find GSUtil, which is the Google command line tool, at that link, the Google Cloud SDK link there. Also Google Developers Console for those people that are sort of managing accounts. Also, you can get account information there too, how much, what charges you're racking up. Docker Hub, there's a link to that. And then lastly, the WDL user guide. The WDL is really well documented, so if you want to look at that, it's very well put together. Oh, right, here's our forum. I think we talked about that. And lastly, this whole piece has been a huge collaborative piece of work across many teams. There's our core team you see here. DISD and KDUX have a large team of people working on it. We've got Alex here, and I see we also have Bradley over there. So quite a number of people up there working on it. Our security team is FISMA and FedRAMP, so been a lot of work from the security team, and we also have UC collaborators that are working on an Atom Spark implementation of FireCloud. That we'll be testing that soon, though I can't predict when we'll roll that out. And we're going to end early. But any questions, come let us know. And thank you. Thank you for your patience, and thank you for guinea pigs. You figure out how to do this.